Mendel's law of segregation states that during gamete formation, the two alleles, or the two traits as he called them, for every character will separate, one going to each gamete. We now know that it's alleles for each gene, so that during meiosis, when the one cell splits to become two and those two split to become four, each resulting daughter cell, which is haploid, gets one allele. Mendel took this a step further. Law of segregation also has with it the law of independent assortment. In his day, before we knew what DNA was and chromosomes and all that, he discussed how one character with two traits, when those two traits separate to go into individual gametes, how they segregate has absolutely no effect on any other characteristics, any other traits. Modern day parlance, as we saw in chapter 11 with meiosis, how chromosome 1 divides, goes through, divides. You know, the tetrads in meiosis 1 separates, and then the two sister chromatids, how they separate, which way they go for chromosome 1, maternal and paternal, along with the sisters, has absolutely no effect on how chromosome 2, 3, 4, 5, all the way through the end. That's what independent assortment means. Early on, when he was looking at the monohybrids, remember mono means singular, hybrid means hybridization, the breeding. You know, he came up, he noticed, hey, look, you know, there's a dominant recessive. Recessive disappears in the F1, reappears in the F2s, but it's at a 3-to-1 ratio. He called this monohybrid crosses. He also, in his, I mean, the guy is brilliant, okay? Crazy, stupid, smart. He also cataloged not only singular characteristics, but he traced multiple characteristics for each plant, so that he could sit there and trace through two characters: height, flower color; pea color, pea texture, or pea shape. He called these dihybrids. Di meaning two hybridizations. He's following two traits. Also could do this for trihybrids, tetrahybrids. You could do the, you know, on and on. Notice there's a big difference when it comes to the Punnett squares. When you're looking at only one thing, even though we'll get to what the artist is talking about here in a minute. If you're following only one thing, a monohybrid, it's a 2x2, okay, because there's only two possible outcomes for the gametes. What we find, though, is it gets a bit more extensive when you're looking at multiple genes. So when you look at, you know, big Y, big Y, big R, big R. Why do we sit there and say there are 4 possible outcomes? Well, let me show you. If we start with this Y can, through meiosis, pair up with that R, or can pair up with that R, because remember, every gamete's going to get both genes. The second Y, this one, can pair up with that one, or can pair up with that one. So if you count through, there's big Y, big R, big Y, big R, big Y, big R, big Y, big R. 4 possible outcomes. So it's going to be 4 across the top, 4 down the side. Even if you look here where it's big Y, little y, big R, little r. 4 possible outcomes. 1, 2, 1, 2. So you count those up. 1, 2, 3, 4. Now, if you're looking at 3, then it's going to be, God, I can't remember. I think it's like 8 possible. 4, and you just get the number gets bigger and bigger. So, 2, sit there and do a Punnett square with 4 possible outcomes. 4 by 4 means you're going to have a 16 square Punnett square. Now let's get back to this experiment Mendel was doing. Mendel wanted to know if the characters, if the traits are linked together. Will you always have to have yellow round? Will you always have to have green wrinkled? Can they be separate? Are they separate things? If they are joined together always, then the possible outcomes, he did the cross, yellow and green gives you an F1 that's all yellow round. If yellow round always has to go together no matter what, then his potential outcomes are going to be his 3-to-1 ratio. Even though it's a dihybrid, he's following 2 traits, is it still going to have the 3-to-1 ratio? Or is it going to be something different? Are yellow and the Y and the R separate? If they're separate, you're going to get 4 different outcomes in the gametes. Well, he did the cross. He did the dihybrid cross. And what he ended up with in his F2 generation was, well, a myriad of different things. He had, just like he did in the parentals, yellow round, and he had green wrinkled. But he also had something new. Green round, yellow wrinkled. These showed up nowhere else. When he counted them up and he did the ratios, he came up with what was a 9-3-3-1 ratio. For every 1 green wrinkled, like he had in the parental, he had 9 yellow rounds, just like he had in the parentals. But he also had 3 and 3 of green round, yellow wrinkled, something new. So this told him that what we now call genes, what he called characters, for P color and P shape or texture are completely separate from each other. One, color, does not affect shape or texture. So we now refer to these as the Mendelian Ratios. If you're doing a monohybrid cross, you're going to have the 3-to-1 ratio. 3 dominant phenotypes for every 1 recessive phenotypes. If you're doing a dihybrid, 9-3-3-1. 9 double dominants for every 1 double recessive. And then you get these mix. For every one double recessive, you're going to have 3 that is a dominant recessive and 3 recessive dominants.